So, continuing our discussion on insertional inactivation and electroporation in plasmid selection system. After a brief incubation to allow expression of the antibiotic resistance genes, the cells are plated onto a medium containing the antibiotic. As an example, let us take it as ampicillin. Now, colonies which grow on these plates must be derived from cells which contain plasmid, since this carries the gene for resistance to ampicillin. So, at this stage, it is not possible to distinguish between those colonies containing plasmids with inserts and those which simply contain recircularized plasmids. So, to achieve this, the colonies are replica plated. So, here we use a sterile velvet pad onto which the plates containing tetracycline in their medium. Since the BAMH1 site lies within the tetracycline resistance gene, this gene will be inactivated by the presence of insert, but will be intact in those plasmids which have merely recircularized. Thus, the colonies which grow on ampicillin but not on tetracycline must contain plasmids with inserts. Right? Since replica plating gives an identical pattern of colonies on both sets of plates, it is recognized as straightforward to identify the colonies with inserts and to recover them from the ampicillin plate for further growth. So, this figure illustrates the importance of second gene for antibiotic resistance in a vector. So, this is about the replica plating to detect recombinant plasmids. So, as you can see in the figure, a sterile velvet pad is pressed onto the surface of an agar plate, picking up some cells from each colony growing on that plate. The pad is then pressed onto a fresh agar plate, thus inoculating it with cells in a pattern identical with that of the original colonies. Now, clones of cells that fail to grow on the second plate, example, owing to the loss of antibiotic resistance, can be recovered from their corresponding colonies on the first plate. Right. Moving further, we have another plasmid cloning vector, that is PUC. The valuable features of PBR322 have been enhanced by the construction of a series of plasmids which we term as PUC, that is produced at the University of California. If you carefully observe, there is an antibiotic resistance gene for ampicillin and origin of replication for E. coli. In addition, the most popular restriction sites are concentrated into a region which we term as the multiple cloning site, or MCS. Moreover, the MCS is part of a gene in its own right and codes for a portion of a polypeptide called beta galactosidase. Right. So, when the PUC plasmid has been used to transform the E. coli, that is host cell E. coli, the gene may be switched on by adding an inducer. Now here the inducer is IPTG, that is isopropyl beta D thiogalactopyranoside. So in its presence, it causes the enzyme beta galactosidase to be produced. Now the functional enzyme is able to hydrolyze a colorless substance. We call it as XGAL, that is 5-bromo 4-chloroindole 3-beta-galactopyranoside. So this substance called XGAL into a blue insoluble material. Right. However, if the gene is disrupted by the insertion of a foreign fragment of DNA, a non-functional enzyme results which is unable to carry out hydrolysis of XGAL. So what is essentially we are trying to understand is, there is an inducer, that is IPTG. When the PUC plasmid has been used to transform the host cell E. coli, the gene may be switched on by adding the inducer IPTG. So its presence causes the enzyme beta galactosidase to be produced. So the functional enzyme is able to hydrolyze a colorless substance called XGAL into a blue insoluble material. Okay. Now, if the gene is disrupted by insertion of a fragment of DNA or the gene of interest, now a non-functional enzyme results which is unable to carry out the hydrolysis of XGAL. So what is the outcome? Thus a recombinant PUC plasmid may be easily detected since it is white or colorless even in the presence of XGAL. Whereas an intact non-recombinant PUC plasmid will be blue as its gene is fully functional and not disrupted. Right? So this system, this elegant system is called as blue-white selection system, which allows the initial identification of recombinants to be undertaken very quickly and has been included in a number of subsequent vector systems. Right? So this selection method and insertional inactivation of antibiotic resistance genes, they provide information on the character of the DNA insert and the status of the vector. To screen gene libraries for a desired insert, hybridization labeled gene probes are also essentially required. So this is basically about the plasmid selection system, and we saw two examples for the plasmid based vectors. Now we will continue to understand about virus based cloning vectors. A useful feature of any cloning vector is the amount of DNA it may accept or may be inserted before it becomes unviable. So, inserts greater than 5 kilo base pairs in size increase plasmid size to the point at which the efficient phages, that is the bacterial viruses, they have been adapted as vectors in order to propagate larger fragments of DNA in bacterial cells. So, cloning vectors derived from one bacteriophage are commonly used since they offer approximately 16-fold advantage in cloning efficiency in comparison with the most efficient plasmid cloning vectors. So, this is a very good advantage. A 16-fold, approximately 16-fold advantage in cloning efficiency in comparison with the most efficient. You can take any plasmid cloning vector, but the most efficient plasmid cloning vector. So, when it is compared, so the virus based cloning vectors, they show 16-fold advantage in cloning efficiency, right. So, if you just look at the bacteriophage lambda life cycle, it is a linear double stranded phage approximately 49 kilo base pair in length, and it infects E. coli with great efficiency by injecting its DNA through the cell membrane. Now, in the wild type lambda phage, the DNA follows one of the two possible modes of replication. First, in the first possible mode of replication, the DNA may either become stably integrated into the E. coli chromosome, where it lies dormant until a signal triggers its excision. This is termed the lysogenic life cycle. Okay. Now, alternatively, it may follow another life cycle which is called as lytic life cycle, where the DNA is replicated upon entry to the cell. Phage head and tail proteins synthesized rapidly, and new functional phage is assembled. Right. So, the phage are subsequently released from the cell by lysing the cell membrane to infect further E. coli cells nearby, so that you produce more number of phages. We just look at the genome. At the extreme ends of the lambda phage are the 12 base pair sequences which are termed as COS sites. That means cohesive sites. Although they are asymmetrical, they are similar to restriction sites, and important function is they allow the phage DNA to be circularized, right. So, the extreme ends of the lambda phage contain a 12 base pair sequences which we term it as COS sites or cohesive sites. Though they are asymmetrical, they are similar to restriction sites, and also they allow the phage DNA to be circularized. Now, phage may be replicated very efficiently in this way, thus resulting in concatamers of many phage genomes which are cleaved at the COS sites and inserted into newly formed phage protein heads. Right. So, the lambda phage has been used extensively in the production of gene libraries, for a simple reason, because of its efficient entry into the E. coli cell, and second thing, the fact that larger fragments of DNA may be stably integrated. So, these are the two important features that makes the lambda phage to be extensively used in the production of gene libraries. Number one, because of its efficient entry into the E. coli cell, and number two, the fact that larger fragments of DNA may be stably integrated. So for the cloning of long DNA fragments, say for up to approximately 25 kilobase pair, much of the non-essential lambda DNA that codes for the lysogenic life cycle is removed and replaced by the foreign DNA insert. Right? I'll repeat it. For the purpose of cloning of long DNA fragments, say for example a maximum of 25 kilo base pair, much of the non-essential lambda DNA that codes for the lysogenic life cycle is removed and it is replaced by the foreign DNA insert. Now the recombinant phage is then assembled into preformed viral protein particles, and we call this process, or this process is known as in vitro packaging. So these newly formed phages are used to infect bacterial cells which have been plated out on agar medium. Right. So this process is known as in vitro packaging. So once inside the host cell, the recombinant viral DNA is replicated and all the genes needed for normal lytic growth are still present in the phage DNA that has not been removed. And so multiplication of the virus takes place by cycles of cell lysis and infection of surrounding cells, giving rise to plaques of lysed cells on a background or lawn of bacterial cells. The viral DNA, including the cloned foreign DNA, can be recovered from the viruses from these plaques and analyzed further by restriction mapping and agarose gel electrophoresis. If you just carefully look at this image, you will find two types of vectors: insertion cloning vectors and replacement cloning vectors. In general, two types of lambda phage vectors have been developed. Number one, insertion cloning vector, and number two, replacement cloning vector. Now the lambda insertion vectors and lambda replacement vectors, they are the two types of lambda phage vectors that have been developed for the purpose of cloning of the foreign DNA. If you look at the features, lambda insertion vectors accept less DNA than the replacement vectors, since the foreign DNA is simply inserted into the region of the phage genome with appropriate restriction sites, and a simple example is lambda GT10. In contrast to insertion vectors, in a replacement vector, a central region of DNA which is not essential for lytic growth is removed. That is, we call it as a stuffer fragment. How do you do that? By a double digestion with enzymes such as EcoR1 and BAMH1. Now the outcome is, as you see in the figure, this leaves two DNA fragments, and we call these DNA fragments as right arm and left arm. Now the central stuffer fragment is replaced by inserting foreign DNA between the arms to form a functional recombinant lambda phage. Right? So the most notable examples of replacement vectors is, it is given over there, lambda EMBL4. And we do have another replacement vector, that is lambda ZAP. right? So this figure is a general map of lambda zap. Lambda zap is a commercially produced cloning vector which include unique cloning sites, and these unique cloning sites, they are clustered into a multiple cloning site, that is MCS. You can see over there MCS in LAGZ gene. Further, the MCS is located within a LAGZ region, thus providing a blue-white screening system based on the insertional inactivation principle. And it is also possible to express foreign DNA, that is foreign cloned DNA, from this vector. So you can also use this vector to express. It is not only transformation, it is also about expression of foreign cloned DNA using this lambda zap cloning vector. This is a very useful feature of some lambda vectors, since it is then possible to screen for protein product rather than the DNA inserted into the vector, because it is possible to express. When there is an expression, you see the product formation, that is the protein. Another feature that make this a useful cloning vector is the ability to produce RNA transcripts, and we term it as cRNA or riboprobes. This is possible because two promoters for RNA, that is polymerase enzymes, exist in the vector, that is a T7 and a T3 promoter. If you just carefully look at the image or the figure, you will find T3 and T7 promoter which flank on either side of the MCS, that is multiple cloning site. One of the most useful features of lambda zap is that it has been designed to allow automatic in vivo excision of a small 2.9 kilobase per colony producing vector, that is the Phage-Mid P BlueScript SK. And this technique is sometimes called single stranded DNA rescue, and this occurs as a result of a superinfection process where helper phages are added to the cells which are grown for an additional period of approximately 4 hours. Right. Now, if you just look at this figure, you can understand how the helper phage is involved in the superinfection process. The helper phage displays a strand within the lambda zap which contain the foreign DNA insert, and this is circularized and packaged as a filamentous phage similar to the M13. Now the packaged Phage-Mid is secreted into the medium from the E. coli cell, and it may be recovered from the supernatant. So thus the lambda zap vector allows a number of diverse manipulations to be undertaken without the necessity of recloning or subcloning foreign DNA fragments. Is that clear? So if you carefully observe the figure, there is a DNA inserted into the MCS, and then you have the Phage-Mid vector site, infect the E. coli, add the helper phage, excision of Phage-Mid. So this is about single stranded DNA rescue of Phage-Mid from lambda zap. That is, the single stranded Phage-Mid BlueScript may be excised from lambda zap by addition of helper phage. So this provides the necessary proteins and factors for transcription between the two sites, that is I site and T sites, in the parent phage to produce the phagemate with the DNA cloned into the parent vector. Right. So, moving further, we have the M13 and phagemate based cloning vectors. Single-stranded bacteriophage vectors such as M13, so these have been a great value in molecular biology. They are all single-stranded bacteriophage vectors. And in addition, vectors which have the combined properties of phages and plasmids, we call it as phagemids, are also an established and frequently used cloning vectors. Speaking about M13, it is a filamentous coliphage with a genome composed of single-stranded circular DNA. Now, what happens during the process of infection? Following infection of E. coli, the DNA initially replicates as a double-stranded molecule, but subsequently switches production to a single-stranded DNA. Now, the resulting virions for infection of further bacterial cells are then synthesized. So, the nature of these vectors makes them ideal for techniques such as chain termination sequencing, for the reason that the chain termination sequencing or maybe even in vitro mutagenesis, both the methods require single-stranded DNA as a source. So, these vectors, especially M13 vectors, are very much ideal for chain termination sequencing methods as well as in vitro mutagenesis assay, because both the methods require single-stranded DNA. So, I'll stop my discussion over here. We can continue our discussion on M13 and other cloning vectors in the next session. Thank you very much.